Good morning, students. Today we are going to study another topic from the applications of botany, and that is plant tissue culture. Tissue culture is a technique of growing cells or tissues or organs or whole organism in vitro condition, and care should be taken that there is a aseptic condition. Haberland in 1902 firstly tried for the culture of isolated mesophyll cells, but unfortunately he did not succeed. But Guha and Maheshwari in 1966 tried for the culture of pollens. Surprisingly, Mascarenhas in 1991 induced flowering in bamboo plants by tissue culture technique. Haberland first succeeded tissue culture into the plants, and Alexis Carrel tissue culture in the animals. But commercially this tissue culture is succeeded in the orchid plants. Here this petri dishes with the young cymbidium orchids culture you can observe, but artificial medium of MS, that is Murashige and Skug, really took off the tissue culture commercially. This tissue culture is based on the principle of totipotency. This is the ability of cell to differentiate and develop into the whole plant when given the correct conditions. Important, that is correct condition. This is because each and every cell is with genetic potential of the parent plant, as we are looking like our parents. Two types of cells are used for this tissue culture: meristematic cells, these are undifferentiated cells at shoot and root tips, and another one, that is the parenchymatous cells, these are the thin walled cells, and they also induce to divide and differentiate. Then important step is to choose the explant. Then we must know what is explant. Then this is multicellular tissue fragment and obtained obviously from the living plant. It is leaf, stem, root, petiole, hypocotyl, or it may be cotyledon, embryo, and it can be a meristem. This is a multicellular tissue. For successful tissue culture outcome, there is a need to prepare the specific artificial nutrient medium, or this is also known as the plant tissue culture medium. It is composed of macronutrient, micronutrient, vitamins, amino acid, source of carbon in the form of sugar, undefined organic supplement, and the growth regulators like auxins and the cytokinin, and lastly solidifying agent as a agar, and this is obtained from the gracillary algae. Here this table is with comparison of the three different media like Murashige and Skug, Hoagland and the White for the preparation of stock solutions of macronutrients and the micronutrients. Single medium cannot be used for all types of plant and organs, because each and every cell is with different genetic mechanism, that already I have told you in last slide. Commonly used medium is a MS medium for the tissue culture, and it is composed of inorganic salt, iron, vitamins, amino acid, plant hormones, and the carbohydrate supply in the form of sugars. So these salts, majority macro and micronutrient salts, are with different concentrations. So stock solution we have to prepare for the macro as well as the micronutrient. Iron is supplied in the chelated form of FeEDTA, and vitamins commonly used the mesoinositol, thymine, nicotinic acid, and pyridoxine, and commonly used glycine is as a amino acid, whereas carbohydrate is in the form of sucrose and general concentration is 30% for the tissue culture. Important aspect, that is use of phytohormone. No doubt they are in need of a very small concentration, but they are important for the success of this growth of tissue in vitro condition. Generally used auxins are IAA, that is indole acetic acid, NAA and the IBA, whereas the cytokinins which we are using for the induction of root, and they are kinetin, BAP and the zeatin. They are used in nutrient medium. Depending upon the use of agar, three types of basic mediums are there. One is solid medium, where we are using 6 to 8% agar, semi-solid medium it is with less amount of agar, and liquid medium it is devoid of agar and commonly used for the cell suspension culture. Many of the times complex additive from the natural sources like coconut milk or watermelon juice also used for this tissue culture media. So for preparation of these different stock solution, we have to mix together in appropriate quantities, and this is your basal medium. But it is not feasible to weigh all the constituents, so we have to prepare the stock solutions of the macro and the micronutrients, so that we can use them in different combinations of the nutrient medium. We can use different stock solution and different concentration, so we can compare their result. For cleaning of glassware, which is important aspect of the success of tissue culture, there is a need of a step-wise sterilization of glassware, in which first we have to soak them in in soap water, then in concentrated HCl, rinsing with the tap water, and afterwards rinsing with distilled water, and finally drying in oven, and we can keep them at oven for 140 to 160 degrees Celsius for 2 hours. After preparation of media, it is important to note its pH, so we can use pH paper, or nowadays these are available digital pH meter. Then the next step is to sterilize that media, and for that we are using this type of instrument, that is autoclave. These are the two different sized autoclaves are there, small and large scale autoclaving of the media as well as the glasswares. Why there is a need to sterilize the media? Because culture medium support the growth of microbes, and sterilization can kill that microbes. They come from any media, that is either it is from nutrient medium, instrument, glass vials, or even from the plant material. So for this, surface of plant tissue as well as the non-living article including nutrient medium must be sterilized. Sterilization of non-living article, it is majoritarily by autoclaving them under the steam of the 15 lbs pressure and the 121 degrees Celsius temperature for the 15 minutes. So that autoclave is with the same principle of the pressure cooker. By maintaining this temperature and this pressure, we can sterilize the glasswares as well as the media. But thermolabile compounds like enzymes, they are sterilized by passing through the bacterial filters, and they are added in the nutrient medium after sterilization in a laminar airflow cabinet. Then sterilization of plant material is also important, as this can be with microbes. So firstly we have to rinse them with the water, then liquid detergent. In lab level Tipol is used. This is mild detergent. So for this 10% solution for 10 to 15 minutes we have to soak this tissue in that. Then afterwards 70% alcohol for 1 minute. in front of laminar airflow cabinet and treatment with the HgCl2 or the sodium hypochlorite that can remove the microbes from your plant material, that is nothing but the explant. So this is what I want to today discuss with you about the basic principle of the tissue culture, that is tutti potentia. Then preparation of media, which is composed of micronutrient, vitamins and hormones, along with source of carbohydrate, that is sugar, and solidifying agent. And the last, that is the use of our nutrient medium and its sterilization. In next lecture we will go for the another steps for the success of this tissue culture. Thank you.